Welcome to the Dinosaur George podcast, a show about paleontology and other earth sciences. Dinosaur George is a public speaker, author, and TV host with 30 years of study in paleontology. He has performed live in over 4,500 events across the US and Canada. Now, here is Dinosaur George.
Well, hey there, everybody. It is great to be back. This is a very special edition podcast. We're calling this episode 105.2. Let me explain why. In this particular episode, we are not going to follow the normal format that we do. We're not going to do any Ask Dinosaur George questions. We're not going to do any of that stuff. And the reason for that is because this is a follow-up episode to episode 105. Now, in episode 105, we had a chance to hear Dr. Michael Pittman from the Honk Kong University tell us some amazing research he had done on the tail quills of a little dinosaur called Psittacosaurus. At the conclusion of that show, he mentioned that he was involved in another study about the color of a dinosaur, but at that time he couldn't tell us anything about it because that announcement had not been made. Well, a few days ago, paleontologists announced their findings of the color patterns on Psittacosaurus. Very exciting stuff. And it turns out that the specimen that they were studying is the same specimen that Dr. Pittman told us about when it came to the tail quills. So he hinted about this new announcement, but he didn't tell us it was part of the very specimen he was studying. So since that official announcement was made, just like I said a few days ago, Dr. Pittman was nice enough to come back on board and tell us all about his role in that new discovery. So now we get a chance to hear how lasers were used to help solve some of the questions about dinosaur color and shading. So let's go into that interview. In episode 105, we were discussing how new technology has given paleontologists this amazing detailed information about the tail quills of a specimen of Psittacosaurus. At the end of that interview, our expert hinted about a new scientific publication that would, in his words, make a splash. Well, it has indeed made a splash, and the whole world is talking. So back with us to tell us about his involvement with this new information is the head of the Vertebrate Paleontology Laboratory in the University of Hong Kong, Dr. Michael Pittman. Dr. Pittman, welcome back again. Great to have you with us.
Thanks, George. It's great to be back talking to you again.
I want to share something with you that I know is important. At the end of our conversation during the Psittacosaurus tail bristle episode, yeah, you had made the comment about how you hoped that it might encourage young people to look at paleontology as a career. I got an email.
Yeah, absolutely.
I got an email from a lady from Massachusetts here in the States who said that her son, he was 9, was so intrigued that he asked her to show where Hong Kong was because he wanted to go there. Now, I think about the impact. Oh, wow. Here's the best part. He named you Dr. Laserbeam.
Dr. Laserbeam. Maybe I should trademark that. It sounds catchy. Well, that's great. Well, if he makes it over, I'll definitely show him around the lab. That'll be great. Yeah.
Well, she told me that he was absolutely serious about wanting to take a vacation there. That he wanted to go meet the Dr. Laserbeam man. So not only did you make an impact, but you got a superhero name.
Exactly.
So this new paper comes out about the same Psittacosaurus specimen that you studied for, to do your study on tail bristles. And you had mentioned that another study was happening. So tell us about your involvement in this new, what this new announcement was and what your involvement was with it.
Sure. So like Psittacosaurus, as we mentioned, like in the last episode, there are so many specimens. There are like hundreds, ranging from hatchlings all the way up to fully mature individuals. And the specimen that we did, the tail bristles, the same one that I'm going to talk about again today, is one of the biggest ones. But what makes this specimen unique is how well preserved it is. It preserves the soft tissue, all over the body. It's a truly exceptional specimen. If there was a top 10 list for fossils, which is quite difficult to compile, I guess, it would definitely be on that list. So we as part of some, I guess, reevaluation or kind of more detailed work on this specimen, we wanted to look at lots of different aspects. So the original paper came out in 2002, but that was quite a brief paper. So the reason I got involved in the Psittacosaurus in the first place is actually by chance. I worked with my colleague Tom Kay at the Foundation for Scientific Advancement in Arizona on this kind of laser imaging method. And, we wrote a paper on how it works and, how paleontology can benefit it, how it can potentially revolutionize different areas of paleontology. And as part of trying to do that, get people interested to make it the standard tool that you should always use when you look at fossils, we got talking with lots of different paleontologists. And so at a conference in the United States, our annual kind of paleontology conference called the Society of Vertebrate Paleontology annual meeting, I got talking with a colleague called Professor Jakob Winter, who's in the University of Bristol in the UK. And he was really interested, and he said, we should definitely do something. And we kept in touch. And then he's like, Mike, I'm working on the Psittacosaurus with a colleague in Germany, a professor of birds called Gerald Mayer. And he's like, well, actually, Mike, you're going to be in the UK anyway. Why don't you come down to Germany and scan this fossil that we've been working on? So I went with Tom, and we traveled all over Germany and made a pit stop in Frankfurt, where the specimens lives, and we scanned it. And at that point, before we scanned it, they were explaining what they were doing. They were looking at the tail bristles in more detail to see if they could extract any more information. They were looking at the skin in more detail because at that point they already knew that there was some kind of color kind of patterns because there are darker and lighter parts of the preserved skin. But they were delving into it in more detail. And basically, in both cases, we used a laser and got these really exceptional results. So in the last episode, we talked about the bristles and how you can see all these details with the laser. Well, with this second paper, Jakob was like, well, actually, we're looking at a specimen. We think that Psittacosaurus had counter shading. And that means that the top of the animal has a darker color than the underside. And you see this in lots and lots of animals today. You see it in sharks. You see it in animals that live in the savannah. And the reason they have this is for 3D camouflage. So if you can imagine a gazelle on the savannah getting shone on by all the strong direct sunlight, where's the shadow going to go? The shadow is going to be on the underside of the animal. So, and the bright spots where the sun's shining is going to be on the top. So by having a dark top and a light bottom, you're actually counteracting the distribution of the shadow when the animal's under the sunlight. And that makes it really difficult for other animals to make out its 3D shape, which is a crucial part of being able to identify an animal, whether to eat it or whatever.
Right.
So we found this in Psittacosaurus.
See that? I guess it's not amazing that color patterns that we see in the animal kingdom today would be different than those before. So it's not the fact that that it was found on the animal. What is amazing to me is that it was preserved enough to be identified. Yeah. That's the fascinating part. I just
exactly. Because it makes so much sense that you would have this, but, because you see it in so many different groups. I mentioned sharks and gazelles. It makes sense that you'll find it back then. But what's being preserved is a pigment called melanin, and that's what gives our skin, our hair, our eyes their color. So this material, because it's a very tough biological chemical, it preserves really well unusually well in the fossil record, whereas other things, other kind of chemicals produced by animals are not as easy to preserve. So, for example, while we have the melanin and can see countershading in Psittacosaurus, we don't yet have, for example, dinosaur DNA that every single paleontologist can agree existed. Yeah.
Well, you think about it. In today, we're looking at brains and internal organs and feathers and color and structure of bristles. It boggles the mind. I've got to wonder. I'm going to stop watching Jurassic Park and I'm going to wait for the park to open so I can buy my ticket.
Well, that's the thing. I mean, like, I always used to think, well, humans are very good at creating stories and, science fiction, creating like Star Wars and all these kind of fantastic stories and stuff. But actually, like, the more I work in the field, the more I'm like, well, actually, a lot of our inspiration, I think Mother Nature does a much better job. So, like, we're really excited with the laser technique because, so far we've, seen the bristles and things in this countershading study. The reason why, so obviously, I mentioned I went over to Frankfurt. So Jakob told me this. He's like, oh, this is what we have. And I was like, oh, this is amazing and stuff. But, there are parts of the fossil. There was one part in particular, the belly region. And the belly region, you can see little bumps in the surface of the skin. And what they are little stones that are preserved underneath the skin in a clump. And those stones are called stomach stones or gastroliths. And modern birds, not all of them, but a lot of them, they ingest stones and they put it in a special part of their digestive tract called the gizzard, and they use that to help them process their food. So this Psittacosaurus basically has that, and in that region of this, the stomach, we have these stones under the skin. There's a hole where in the kind of back of the how should I say this? The back end of the stomach region. So if you're looking at the underside of the animal towards the tail, that region's really pale. But if you go kind of towards just before the chest, it's actually quite dark. And when I looked at it, the specimen, I was like, well, wait a minute. If this is countershading, then that area really should be light as well. And he's like, well, the hypothesis that we have is that part of the region, the skin seems to have been punctured. So some of the melanin from inside the animal, because melanin actually helps to pigment organs like the liver, might have leaked out and basically kind of covered everything in melanin and kind of masked it. So this is where the laser came in, because I was like, well, okay, we can test that. So we shone the laser at it, and what happened when we processed the image and looked at the final result, that ink on the pigment on the top of that we thought was just on the surface and not in the skin, the laser penetrated through it and revealed the pale stomach area.
Oh my gosh!
Yeah.
Oh.
So we were like, wow, because you imagine, like, because if it's like spilling some kind of cranberry juice on something, it's that really tiny veneer of material, right? And the laser managed to destroy, and it really blew us away because this is a cutting edge technology, we're still trying to kind of feel the edges and find its capabilities. So when this happened, we were like, wow. So immediately when the guys saw the results, Joe wasn't with us at the time, but Jakob wasn't with us at the time, but Professor Mayer in the Senckenberg, this museum in Frankfurt, he was like, wow, you guys have gotta get in, help us with be involved in our study. So it ended up being like a beautiful kind of synergy and collaboration, because none of us were expecting what we found, and to basically help support their work further, it was really great.
God. So had you already done your test with the tail bristle part before this, or did that study come after this?
Well, the data collection, we actually did. We took a picture of the whole animal. We did some close-ups. We did zoomed out shots. We actually did it over 3 or 4 days in one trip to Frankfurt. And the first thing that popped out was actually the tail bristle stuff, because that was the thing that the professor that was with us, Professor Mayer, he was working on a manuscript that he was kind of in the full swing writing, and he kind of wanted that image. So we looked at those images first, found that they were great, and then within those 3 days looked at the other images, and it was just amazing.
It's so fascinating. You say that with such like, yeah, the tail bristle was amazing. Then we found this, and I'm sitting there going, oh my, I can't get past the tail bristle part. And this, and this is just unbelievable. So with the laser, you literally could look through the outer layers to see what's underneath. And that is sort of what helped them also discern what was true color of the skin versus what was coloration that may have been from beneath leaking out, right?
Well, yeah, like, for example, you know how in the movies, I'm not exactly sure how they do it, right now, but like in the olden days, they would have, like, I don't know, some dye in the person's shirt, and then they would, pretend to shoot the person, and the dye would leak out. So if you imagine you've got your Psittacosaurus with, its organs inside, some of them got punctured, and then the skin got punctured, and some of that leaked out and basically made a mess over the top of the animal, over the underside, so kind of completely covered it. So this animal's kind of preserved in a heap and got buried obviously in the ground. But when it was in that heap, some of its internal organs kind of leaked out over the skin, so it wouldn't have been particularly pleasant to look at. So luckily, this thing is a rock, so it doesn't smell, or it's not too. You won't faint if you look at it, and but yeah, basically, yeah, it's just this tiny veneer. It's just like this, essentially this liquid fluid kind of came out onto the skin, and the laser, and then eventually that material was eventually preserved as a fossil, and the melanin part remained, and then the laser was able to basically look past that. So it was really good. So we've been doing a whole bunch of stuff with lots of other specimens, and basically finding similar things. The laser can actually basically penetrate a few millimeters into the rock and help reveal information.
Wow. Let me ask you, is it normal for paleontologists to, like you had said, you doing this with one paleontologist and then you're doing something with somebody else, is that pretty normal that you guys work together on various projects that maybe you would have never had anything to do with this particular animal, and yet now you're associated with helping understand the colors, and you're associated with understanding the bristles? Is that sort of thing normal in the paleontology world?
Yeah, I guess there's a whole range of styles that people like to work in. Some people only publish work by themselves. They only ever see John Smith's work, and because John's happy to go about and do his own stuff and get on with it. A lot of people, maybe they have a buddy that they click with, their science mates, soulmates, but they're science mates, and they always publish their stuff. But and then what often happens if you become really specialized in one particular area, and people will seek your expertise. So if you've got some sort of specimen and it has, I don't know, for example, some coloring. So Dr. Winter, he was actually the first, I believe, or at least one of the first, I think the first, who made this association between the feather color, preserving these kind of melanosomes in fossils and reconstructing feather color. So, when people want to work on that, he's often, one of the people that, people, seek out. And then you got others that it's, very different. There's all sorts of different ones. So in our case, we've got this method that, Tom Kay, my colleague in the Foundation for Scientific Advancement, he actually applied this use of laser imaging specifically to vertebrate fossils. And so he's the guy, and, we've been working together, imaging lots of stuff. So if people wanted to do that in the future, then, obviously in that case, we would be the who you got to call people. I think it really varies. And often the way collaborations work, it's really strange. Like, I'm always surprised as well by how different collaboration things, work out and stuff. And often with projects, it just so happened that you were there at the right place at the right time. So, for example, I've been visiting a museum, for example, to look at one particular specimen for for a specific project. I met someone who's also looking at the museum, who's working on something else. And then they'll be like, well, actually, Michael, what do you think of this? And then you give your opinion or whatever, and they'll be like, oh, wow, I didn't realize that. And, and maybe you gave them a really good idea and like, wow, that's really good. We should roll with that. So it's science actually, people tend to have their own stereotypes of scientists, but doing forefront science, you really have to be quite creative. And a lot of, as a musician, an amateur musician, I used to play in a blues band, and so I'm quite creative. I write music. Bizarrely, I see a lot of parallels between science and music, the way that you kind of go about doing it. And it's always kind of really great, when your expertise is sought. But I often enjoy the projects that, they just seem to come out of nowhere. So this project at the moment, the Psittacosaurus, was me saying hello to Jakob, who I knew did the feather coloring work, in a hotel lobby in the conference venue. Wow. And kind of just being polite and introducing myself. And 2 years later, we've got this cool paper on Psittacosaurus.
Well, what a cool way, though. I mean, what an amazing thing to think that how the public benefits by the research done and by the connection. Like you said, it's a conversation in a hotel, and without your expertise and knowledge in the use of the laser, would we be talking about this today? Or if we were, would they feel as confident about the result had that not been, had that not been the use of your research to do it? So even though it wasn't your project per se, your participation made it that much better and that much more amazing.
Yeah, I mean, it's sometimes a lot of the discoveries that I've made that my favorites, it kind of completely unexpected. There's only so much you can kind of plan, and so that you can, for example, if you're interested in this area, work towards, and I'm going to do this, and this to achieve that. But a lot of it, it really, you get taken with the current. And at the moment, I'm actually, my lab's the vertebrate paleontology laboratory, but I'm actually working on some really old material from some invertebrates, so they don't even have a spine, with the kind of laser methods. And that was also really strange, because I went to visit a colleague in the UK, and walking up to his office, bumped into one of his colleagues, and she happened to be studying, some stuff that, some fossils that would made of material that would fluoresce. And we got talking, and the same thing's happening again. So hopefully we'll have some other cool discovery. But I enjoy that part of it, because you're not expecting. It's it's kind of in some ways it's kind of high risk because you're like, oh, let's give it a shot. It could be, could be amazing. It could, but I quite like that because you're kind of like, you're really kind of not sure what to expect, and it's great.
Right. So, do you think now that you'll take this technology and apply it to some of the specimens, like, for instance, some of the mummified specimens? Do you think that this technology will be as effective on some of, or are they already lined up to possibly go back and look at some of the other specimens now that you know what you're capable of doing?
Yeah, absolutely. So this Psittacosaurus is from northeastern China, the early Cretaceous, and there are tens of thousands of fossils from those kind of deposits in northeast China. And we've obviously there's a lot to work through, but I can say that from similar rocks as Psittacosaurus, we've probably scanned, I think probably like 1,000 or 2,000 specimen. So we got one manuscript that I just submitted the other day, which is a study. This is a really cool paper. Is just on one specimen, but we've got a paper where we look at hundreds of the same animal, and as you can imagine, if you have hundreds of the same, you have a very nice way of kind of checking that all your inferences are right, and if you've got missing data, if you've got lots of specimens, you've got a really good chance of filling that in. So, we're actually going for kind of something similar with another animal, but I'm unfortunately going to have to keep you guys waiting a little bit. But yeah, this Psittacosaurus is based on one exceptional fossil. But we're kind of super excited and just been scanning stuff like mad. It's been stuff in China, but we've also been over when I was in that Germany trip. We actually looked at some German fossils from like the Solnhofen. So it's kind of late Jurassic, tropical kind of lagoonal environment. So if you think about Barbados today.
Right.
Or somewhere nice and hot with beaches and stuff. A late Cretaceous ecosystem like that, we've been having some success looking at that material as well, and seeing how that fluoresces. So yeah, we've been keeping busy and focusing on formations, places where you find fossils that are called Lagerstätten. So these are given this funny German name because they're very, very well preserved. So in the United States, there's several, you've got the Green River Formation in Utah. In Canada, there's the Burgess Shale, which are for very, very early invertebrates. We're kind of focusing on those at the moment because those types of rocks already preserve, the best detail you can get with fossils.
Right.
So we're hoping to kind of push the envelope with the laser to see what we can find. So, for example, with the Chinese material, these feathered dinosaurs, we can see skin, we can see feathers. There's even a fossil from Italy, a dinosaur fossil, where you can see the different organs, like the intestines, and that, amazing. Scipionyx is its name. But, the reason we're focused on these Lagerstätten, because wouldn't it be amazing if we could get the laser could show up a heart?
Oh.
Or, yeah, exactly. Or like the throat, or, and so you can help bring these animals to life.
God. Now, is there any evidence so far that the melanosomes would have survived going all the way back to the Jurassic, or going back to like the Burgess Shale stuff, for instance? Is there any evidence that would suggest that one day, with the use of lasers or any technology, we can understand colors even going that far back in time?
Well, the principle just works with any mineral that fluoresces. So calcite is the material that you get in caves. That limestone's made of. That if you shine stuff with a laser, glow, fluoresces really, really brightly. And in these, the Psittacosaurus, for example, what's actually fluorescing is basically material called like apatite. So that's a very kind of common mineral that you get in rocks. And so as long as they have those things, then these fluorescing minerals, then you can go all the way back. So this stuff, I can't reveal too much, unfortunately, but this stuff, this colleague of a friend that I bumped into, and decided to laser some of her fossils. Those are like 500 million years or kind of thereabouts. So you're much older, and it's still working because they have these kind of minerals. So really, it's we're pretty excited because it's we really don't know what to expect, and there should be some nice discoveries to make.
God, you are the most interesting and yet frustrating person to interview in the world. Because you sort of wave this little flag of, hey, here's what's coming next, and then you simply say, but I can't talk about that right now. You should you should be in politics.
We have that. That's the problem all the time with we like, you go to a conference, you're like, oh, what have you been working on? And I'm like, I really want to tell you what you it's kind of like, but yeah, it's so no, it is nice. Like for this Psittacosaurus, we kind of I've been working on it for, Jakob and his colleagues at the University of Bristol working at it for a lot longer, but like at least I think I've been involved in that since like about March or April, so the last few months. And I've just been like, oh my god, I can't tell anyone.
Well, what is exciting to me, I find to be the most exciting, is that the color patterns were what we see in the animal kingdom today. And one of the things that I think we were going through a trend, and maybe still going through it, where a lot of the art had gone so far over the top with these remarkable, outrageous color patterns, which they're definitely in the animal kingdom. But when I would look at some of it, my reaction would be, I would be able to see you from 10 miles away if I'm a predator. And although, gigantic orange blotches look amazing, is it really that effective if you're trying to hide from somebody you want to eat, or you're trying to hide from who's trying to eat you? And so I'm kind of excited that the color patterns are not alien, but they're more that way. You can tell a child, go to the zoo and look at the colors there. And so the idea of striped hadrosaurs, like a zebra, start to make sense. There would be a reasoning for that. And so this little guy's color patterns are great.
Well, the paleontologists, when we do our work, we're always grounded in what we know from the present. So, even though I'm, digging in the desert for things that were, alive millions of years ago, I'm, dissecting modern animals. I'm, looking at, how modern animals behave, and making use of, what we know about, animals that we can see today, and even actually looking at the finest details. So like looking at molecular pathways, really, really the nitty gritty of how animals work, and kind of applying that kind of information. And a lot of the time, we use that information, but sometimes there's a limit to how much we can actually take and put in, a given situation. So, with the color, for example, you mentioned before, we definitely find this color today, and it's possible, but, is it reasonable or whatever? Like, you can take some information from the present, but how strong is that information? And often, it's frustrating when you've got a cool fossil, and often maybe there isn't anything modern that can really help you to understand what you're seeing. But in the case of the Psittacosaurus, we know they have the counter shading, but what really kind of makes it particularly exciting is this idea of counter shading. There's even differences from about linking the type of counter shading with where the animal lives. So, for example, out on the savannah, where you're in direct sunlight, there's hardly any trees everywhere. If you look at these ungulate animals like gazelles, they have a very sharp distinction between a very dark top and a very light underside, and there's a very sharp boundary. So it's like brown-white, really sharp.
Right.
And that boundary is very high up on the animal because when the sun's mainly coming directly from above and it's direct, it's the shadow takes up quite a lot of the body of the animal. But when you go into forested environments, the animals there, when they have counter shading, that line is not as distinct. It's more gradual, and the transition happens lower down in the animal. And so, in the paper, we take this information from the fossil, made a 3D reconstruction, and did an experiment where we put this animal, well, I say we, but that part of the study was Jakob and Professor Innes, the other colleague at Bristol who are leading that. They put this model in the sunshine, in direct sunlight, in this kind of shaded, diffuse sunlight, and found that the way that they were able to use this information to predict where the transition should be if Psittacosaurus lived out in the open or if it lived in a kind of forested, more shaded area. And the crazy thing is that the pattern we found in the fossil matches animals today with counter shading that live in forested environments. And for us, that was like, whoa.
So it's not just we have a better understanding of the color, but now we have a link between the shading and counter shading of the animal being appropriate to its environment. And I guess the way you could verify that would be then to look at the plant fossils found in association with the animal?
Yep, already done. So we already know that Psittacosaurus was living in a forested environment with a temperate climate, with mainly coniferous trees that you get in the northern parts of North America. So that was the icing on the cake. We already know where it lives, and this color shading shows that corroborates that. So it was amazing.
How exciting is that to to see it all make sense? Because the worst thing would have been is to find that the counter shading would have been reversed and wouldn't have made sense. And then you're left with, do we not understand this animal? But when you see the evidence, it's all taken independently. And when it's all put together again, it falls directly in line with at least having modern animals as a sort of a basis for it. And to match shows you that evolution is always going to be in the best interest of the animal, and animals' colors have perpetuated through all that time, being just as useful today as they were 100 million years ago.
Yeah, exactly. And I'm going to make a prediction because, you heard it here first. Today, if with skunks, they have reverse counter shading. You'll notice that they have white on top,
right,
and black underneath, very sharp boundary. Well, rather than being to disguise the 3D shape of the animal to make it harder to see, the reverse kind of shading, the current understanding is that it's used to basically say, stay away from me, I'm mean and I'm gonna. So it would be cool, if we find more fossils with that exceptional preservation and they are extremely, extremely rare. To find the reverse thing, especially if the animal, had some sort of other anatomical feature which suggests that it, you didn't want to mess with it,
God, how amazing would that be to, I remember there was a there was a time, and I don't know whatever came of it, that somebody had proposed the idea of a venomous
Yes,
dinosaur.
Yeah.
And how remarkable would that be if that specimen or other specimens were so well preserved, like you said, and you would find this reverse counter shading, then you might think, well, this guy's got a walking billboard that says, you don't want to mess with me. So you're right. He doesn't want to be hidden. He wants to be recognizable. I mean, that would be over the top fascinating. So with their relationship, though distant, with reptiles, and reptiles having the ability to alter their skin color, is there any way that you think we would be able to learn something like that? Like, let's say, for instance, your little Psittacosaurus is perfectly ideally suited for his color patterns in his environment, but during a moment of fear or mad or do you think the possibility exists that they could alter their skin color?
Well, I mean, that, if you look at YouTube videos or see these animals in life, like chameleons changing, it's absolutely mind-boggling. You just look at the animal, you're like, whoa. It's actually quite rare if you look at it within reptiles. There's so few animals that can do that. And for example, you have even better kind of camouflage artists, like the ultimate ones are actually cuttlefish. Like if you see videos that people have been this experiments done where it's kind of quite neat, but they basically have the cuttlefish in a little tank, and they have a picture that they put underneath the tank, and they keep replacing it, and you can see how the cuttlefish changes its color to match, and there's been all these experiments to find out how it works. But so they're the ultimate camouflagists. But chameleons are pretty close. They're amazing. And the way that they do that, though, there's not really a osteological correlate, something that preserves in the bone anatomy. And in terms of the kind of soft tissue anatomy, we really know that you can preserve skin and, I mentioned before, intestines. But the kind of level of detail that you need to preserve to be able to, say if these had the kind of structures needed to, have chameleon-like kind of skin color change.
Right.
Unfortunately, we don't have it at the moment. But, that's, but these things, are constantly changing, and people are able to find these kind of bony correlates on the skeleton, or look at, soft tissues and stuff. So with the venomous dinosaur one, that one of the arguments for that, which I think it's fair to say that not everyone's on board with that, but
sure.
One of them is that in the teeth they have these kind of grooves on the sides of their teeth that look very similar to the groove structures that you have in kind of venomous reptiles today, and some other kind of features. But yeah, it's tricky because you need to have even if you have the bony thing, it could actually be associated with something else. It's very, very tricky. So like with this Psittacosaurus example, the fact that you can have all these different things and they kind of correlate with this, and this, that's kind of quite a rare thing, but it's kind of, as paleontologists, what we kind of, that's our holy grail when you get things like that and you're like, wow,
Well, I've got to wonder how many paleontologists in the world are racing back to look at some of their specimens in their collections to find out if they could achieve what's been achieved through your colleagues and yourself. And who would have thought that at any time we would be sitting around discussing the intricate details where just, 20 years ago, that this sort of conversation we never would have thought we would have been talking about the things we're talking about now. Well,
we're realizing that actually a lot of these soft tissue parts of the skeleton are actually not as hard as to preserve as we thought. So to give you an example, in Canada, Canada's like along with USA and China and Argentina, places like that, these kind of key places for dinosaurs. In Canada, they've been finding up there for like the last 100 years amazing dinosaur skeletons. Some of them are beautifully complete. And recently, even though the place has been looked at like 100 years ago, they actually found a specimen in the rock matrix that entombs the bones. They found preserved soft tissue. And it's not as if the geology wasn't, particularly different from all these other hundreds of skeletons that were found in the past. But what people, talking to the colleagues that do this work, I think they just basically suspect that back then they were like, oh, there's all this crud in the rock around the bones. Let's prep that away. Let's destroy it and take out the bones. And I think in a lot of deposits, they're actually like, oh, wait a minute, guys, be a bit more careful.
Yeah.
So, yeah.
Well, you're right. It makes you wonder. It certainly makes you wonder how much had been destroyed.
Well, I can tell you like this. I can tell you this detail because this is cool. And in some northeastern Chinese fossils, so in the Psittacosaurus, you look at the photos of the skeleton, you can see the skin. The laser just makes the detail that you can see, a lot more vivid. You can see better details. But I've been scanning specimens from similar rocks from the same time, and we've just found fossils in a slab that are just bones, and we put the laser in it, and you can see like soft tissue that you can't see with your eye. It just pops out. Yeah. So it's, obviously when you see that, it's amazing. But the depressing part is, not seeing it, well, it's not there.
Right.
You can see the preparation marks that have been made into the soft tissue where a preparator has unknowingly destroyed, beautiful soft tissue because they can't see it.
Right.
So with this laser, we're like, no, stop, that's impossible. So with my colleagues, I'm like, in specific areas where I know that we're getting this kind of soft tissue preservation that you can only see with the laser. I'm like, no, don't prepare it yet. We should image it first, and then you can prepare it. So that's why we're trying to make it a standard technique, because you don't want to destroy the soft tissue, and it's we're hoping that that'll become a reality.
Right. Well, I've often wondered, especially during those times that are known as the Bone Wars, I often wonder how much could have been destroyed in that race to get things out of the ground, and certainly paleontology has changed now to where people are much more careful. But now with research like yours, it's going to be one of those things where you're going to want to be as absolute careful as you can be when it comes to this, because if you find something amazing, you can always take it to Dr. Laserbeam, who will be able to uncover who knows what sort of information. So you're an expert at teasing us on something coming up. Can you wave your little tease to us about anything at all coming up?
Oh yeah, sure. So I've got, in terms of laser, I've got something that hopefully will be accepted, and that's actually this time an animal, a theropod dinosaur. So This animal was a carnivore. So I kind of the stuff that's found, I think it's not color patterns, but I think equally cool, because one of the areas I'm really interested in paleontology and trying to make the laser help solve a lot of unanswered questions is about the origin of birds. So the evolution of birds from the ancestors, from animals that kind of more resemble kind of T-Rex and things. So I'm interested in how that major transition happened. So I've been kind of using the laser to scan lots of key specimens that relate to that. How did an animal that looks more like a T-Rex with a long tail that lived on the ground evolve into, a heavily feathered animal that could glide and eventually fly? And we've been finding so far is details that that help to fill that in. So unfortunately, that's I have to leave you hanging. but for me, that's my kind of personal. Or if I could help and resolve a few things, because a lot of these feathered dinosaurs, they preserve amazing details, and people have done aerodynamics simulations with the raptor dinosaur, Microraptor. It's got feathered arms and legs, and people have modeled how it can move through the air, and they found actually this thing can glide. But that's actually just based on bones and feathers. How about the actual the outline of the body? What were the shapes of the muscles and stuff? That obviously has a huge impact on how the animal behaves and functions. So there's all these missing gaps in anatomical information, and really I'm hoping that the laser could help out that way, and at the same time, a service to the rest of the community. And there's so many other paleontological questions that need to be answered. We're kind of trying to seek out how the laser can really help with those. If you have the earliest life on Earth and stuff, can you imagine how cool would it be if you ended up finding something that you can't see?
Right.
So yeah, it would just be amazing. So yeah.
Well, we're definitely going to stay on top of your work, and I follow everything you do. And again, I appreciate any time you contact me to say, hey, this is now for public announcement, because it is fascinating, and the work you're doing is fascinating. He is a research assistant professor and the head of the Vertebrate Paleontology Laboratory in the University of Hong Kong. It is Dr. Michael Pittman, one of our absolute favorite guests. Thank you again. I realize it's late. What time is it in Hong Kong right now? It's
10 p.m. We've got a big time difference.
Boy, you're not kidding. Yeah, I'm just waking up and I'm just getting started. So you're actually from the future.
Yeah, exactly.
What are today's lottery numbers, by the way?
Yeah, exactly.
Well, I can't thank you enough for taking time out, and especially for doing this on a Saturday night for you. Which I guess that shows what kind of a life you've got. You're sitting at home on a Saturday night talking to somebody in San Antonio. That maybe.
No, it's been my pleasure, George. I really enjoy our chats, and hopefully, if some of these other things, work out, then hopefully we'll have some other cool stuff to report.
Great. Well, keep us in the loop, because we would love to hear it. Thank you again so much for doing this. I really appreciate it. Well, there you have it, everybody. That is a special follow-up podcast. We're calling it 105.2. It is a follow-up to episode 105. I hope you guys enjoyed it. It is incredibly interesting stuff to hear how technology is changing the study of paleontology so dramatically. Think about this. We're at a point in science where we're able to not only see color, but can now understand the patterns of color distributed on a dinosaur. It's something I don't think anybody could have foreseen, and it simply makes me wonder what the future is going to hold. I would suggest to all of you kids to start saving your money, because it looks like Jurassic Park will be opening soon. Unfortunately, we've all seen the ending. Somebody's getting eaten. All right, everybody, that's it for this special edition. I hope you've enjoyed it. I am Dinosaur George.
Thank you for listening to the Dinosaur George Show. Please follow us on our social media links and join our mailing list. If you're interested in having Dinosaur George speak at your event, please visit our website at dinosaurgeorge.com. Until next time, keep digging for clues about the past.